Good day, everyone. So this video is talk about the cytoskeleton. I think this is the first time you heard about the cytoskeleton. So cytoskeleton actually is the skeleton they found in the cytoplasm. So as you see in this diagram, they show a building, a part of a building, and then you can see there are many iron bar. So showing assumes that this is the skeletons of the building. So we go back to the cell. So in each cell, they also have the skeleton. So this skeleton that support or provide the shape to the cell is known as cytoskeleton. The cytoskeletons can only be reviewed under electron microscope. So it is the background, or we call it as a ground substance of the cytoplasm. So there are three types of the cytoskeleton: intermediate filament, microtubule, and actin filament. So the cytoskeleton is used to support the shape of the cell. See, they support the shape of the cell and anchor organelles to fixed location. Okay, for example, you can see this nucleus, ER, okay, and other organelles, for example, mitochondrions, and move organelles. For example, they also can move the mitochondrions here and there, and move chromosomes during cell division by spindle fibers. So spindle fiber is one of the cytoskeleton. Then also move the cell. As you see in amoeba, they can move by using the pseudopodia. So the formation of the pseudopodia is also due to the change of the cytoskeleton in the cell. And also endocytosis and exocytosis. Okay, the cytoskeleton is a dense network of protein fibers. So as you just now I mentioned, there are three types. One is the microtubule, then the second one is actin filament and intermediate filament. Okay. You can see the distributions. This is the microtubules. This one is intermediate filament. This is actin filament. So later you will see what is the function they play by each of these cytoskeletons. Okay, microtubules is the thickest filaments. Fine, long, and unbranched, rigid and hollow cylindrical tubes. So the center is a hollow tube. And then made up of helically arranged subunit we call the tubulins, alpha tubulins, and beta tubulins. So the alpha tubulins and the beta tubulins combines to form a dimer. Then this dimer, you see, they can add in. That means this is plus N, so this is minus N where they're disassembled. Okay, disassembling. So microtubule can be lengthened or shortened by adding or subtracting the tubulin dimers or dimers. Shortening is to move chromosomes. So they serve as track for the movement of vesicle organelles, such as mitochondria, and interactions between microtubules drive the movement of cell. So from here, I think you are quite familiar with these shortenings to move the chromosome by the spindle fiber. So that means the spindle fiber is formed by these microtubules. So in plants, microtubules helps to control arrangement of the cellulose fibers of the cell wall. So microtubules, see, they are found in the centrosome here. So in animal cell, in the centrosome, there are centriole, a pair of centriole. So from the centrosome, see the microtubules, they are forming. Okay, and also the microtubules are involved in the formation of the cilia and flagella. Centrioles is a pair of short hollow cylinders. Okay, cylinders. So this one, there are 27 microtubules. You can see here 1, 2, 3, 1, 2, 3. So they have 9. So we call they all together is 27 microtubules. And these microtubules arranged into a pattern of 9 plus 0. 9 is around, 0 is at the center. Center has nothing, so it's 0. These centrioles that occurs in pair per animal cells, that means in animal cells, and then located in centrosome, okay, that near to the nucleus. Then oriented at right angle to each other. You see, right angle, okay, right angle, 90 degree. Duplicate and separate to opposite cells pore during mitosis to form spindle, the spindle fibers, okay. So the centrosome of plant and fungi lack of centrioles, but they still contain microtubules, microtubules organizing center. That's why in plant cells during cell division, you can't see the centriole because they have no centriole, but they're still able to form spindle fiber because they have a microtubule organizing center, okay, in the centrosome. Centriole also give rise to basal body, okay. So this is the cytoplasm here. This is the basal body of the cilia or flagellums. So then they form the cilia or flagellum of cell. So cilia and flagellum, look at here, they contain 18 microtubules. Okay, they got 18, that means each got 2. So, and then the center, they have 2 microtubules. So this pattern is known as 9 plus 2 pattern. Okay, compared to the centriole, which is 9 plus 0, it's different. Then the cilia and flagella, you already know the flagella and cilia is for movement. Okay, cilia is the movement of mucus or substances, ovum, and the flagella is the movement of a cell. Okay, bacterial cell or and the sperm cell. Okay, so from here you can see this is to show you the sperm cell. This is a chlamydomonas with 2 flagella also for movement. So this is a paramecium that contain a abundance of cilia also for movement. So either the flagella or the cilia, the beating patterns different. Okay, the different. So this one is same, either cilia or the flagella, they share the same, what we call the basal body, and also this one, the what we call the 9 plus 2 microtubules. Okay, so flagella are longer than cilia, so normally found in one or in pair, that means 2. So wave of bending propagate from one end to the other in snake-like undulation. So this one you can refer to the video that I sent to you before regarding the locomotion of sperm. Cilia are shorter than flagella, and then usually they present in great numbers. Okay. So how's that beat is very simple. So they just beat like this way, and then after that return to the original place, and then after that again. Okay, so from here you can see the power stroke here, they move to this side, and then after that they return, recover. Okay, so flagella, they move in different way, in a propeller or corkscrew like this. So like this. Okay, so this is the movement of the cilia, okay, to move the mucus. Okay, intermediate filaments only found in multicellular organisms. Okay, the size is in between microtubules and the actin filament. So what is the functions? To provide cell with mechanical strength or support, and support nuclear envelope. Okay, that's why you see they move around here. Okay, they found around here. Cell-cell junctions, like those holding skin cells tightly. Okay, they hold the cells tightly together. Okay. Microfilaments. So they got 2 types, actin. Here got actin and also tropomyosin. Okay. So what you see here, they are actin filament. Okay, actin molecules. See here they get plus n, so they got minus n. So that means they can lengthening or shortening. So they got 4 major roles. Once maintain the cell shape, the cell shape. That means all the cytoskeletons, they also maintain the shape of the cell. Okay, and then after that they have different role. So for like this one, actin filaments, they have help the entire or part of the cells to move. For example, the amoeba, they want to form the what we call a pseudopodia, so move from one way to another way. So this is helped by the actin filament. So cytoplasmic streaming function is pseudopod, you see, pinch the muscle cell into the two during the cytokinesis, interact with myosin in muscle contractions. So you will study this in next semester, and support for microvilli in intestinal cells. So all this is related to the actin filament. So you can see here, so the movement. So you can see the what we call cytoskeleton, okay, of the actin that moves to this side. Okay, so this is for amoeboid movement. Let's support the microvilli. See, they're around the structure, so they provide the shape here, cell set, and also to support the microvilli. So cytoplasmic streaming, okay, to move substances within the cell, and also during cytokinesis, so they form a pinch of the cell. So this is the movement, okay, the movement of amoeboid movement, okay, of a cell. So actin polymerization, so they move to this side, and after that they disassembling. Okay, so far you can see these three types of the cytoskeleton and each function of this. Okay, you just need to know the type, the structure, and also the function. Okay, so that's all for this video, and then thank you for listening.